The last section of this lecture talks about resistance, and then we'll end by talking about antimicrobial susceptibility testing. So drug resistance. Drug resistance means that the microorganism survives in the presence of the drug. A person does not become resistant to a drug, and that's something that I hear all the time. Even in the healthcare field, when I take my kid to the doctor, I can hear people talking like this. So if I take penicillin every day of my life, it doesn't mean that my body is all of a sudden going to resist the drug. Resistance comes from the microorganism. So the bacterium would develop resistance against the drug. A person does not develop drug resistance. So drug resistance is the survival of the microorganism in the presence of a drug. There's intrinsic and acquired resistance. Intrinsic is the nature of the cell. So the cell would have never been affected by the antibiotic. For example, if you try to use penicillin to kill malaria, it's never going to happen. Malaria doesn't have peptidoglycan. It's going to be completely unaffected by penicillin. Acquired resistance is gained, and usually it's gained by horizontal gene transfer or a mutation. Horizontal gene transfer is the transfer of genetic information from one cell to the next. We learned about that in genetics when we talked about conjugation, transformation, and transduction. So conjugation would be the transfer of genetic material by that little pilus, so bacterial mating. Transduction is the transfer of genetic material by a bacteriophage, and transformation is the uptake of DNA from the environment. But the other way is by random mutation. So here's a picture that shows a skin infection. So here's an abscess, and inside of that abscess you've got bacteria all over the place, and the bacteria will eventually mutate. Mutates happen spontaneously. There's really no reason why. Bacteria have a much higher mutation rate than human cells because they lack a lot of the regulatory mechanisms that prevent mutation or fix mutations and proofread DNA to make sure that these mutations don't exist. So in a cluster, over a period of time, one of them is bound to develop resistance. When you take a drug, it's going to wipe out the ones that are susceptible, to killing by the drug, and it leaves the resistant one. Now, under normal circumstances, if you take all of your antibiotic and you take it as prescribed, it'll wipe out all of these guys. This guy will still be alive, but your immune system will eat it because it's just one cell. But let's say you started taking your antibiotic, which started decreasing the numbers of the ones that were susceptible, and then all of a sudden you started feeling better and you stopped. Well, now about half of these are left, and those half protect the one that's resistant. So now this resistant one starts to replicate, and then you develop into this scenario where you have a lot of resistant ones in comparison to the ones that are susceptible. And then you're like, oh no, I'm still sick, I better take my antibiotic again, and you start taking the antibiotic again. Well, it's not going to work at all. So now you have to go back to the doctor and get a different antibiotic, or you just have to hope that you can clear the infection on your own. So this is how resistance develops. Random mutation, or you have that gene transfer that we talked about. But a couple within a group are going to naturally be resistant. And if you take the drug as prescribed, you should be okay. But if you don't take the right dose or skip a dose or happen to not complete your prescription, then that's when you have problems arise. Low dose exposure, like environmental exposure, there's a lot of hospital runoff that goes into natural bodies of water. So patients in the hospital are taking antibiotics and they're urinating, and that urine goes into the plumbing, and the plumbing goes to the water treatment plant and eventually gets dumped back into natural bodies of water. Well, now that's creating for a mess of antibiotic resistance in environmental settings where you have that runoff from hospitals. You also have people that flush the pills directly down the drain. You have people that dispose of them outside. So in the regular garbage, there's antibiotics being exposed all over the place. You also have farmers that are now giving their animals antibiotics, and that's causing a problem. So we really need to control antibiotics a little bit better. So what are some major resistance mechanisms? How does a microorganism become resistant? Well, number one, efflux pump. Some microorganisms have developed these pumps that pump out the drug. So when the drug comes into their cell, they just pump it right back out. If they pump it out, it has no effect on the cell. Other microorganisms prevent their penetration. So the drug can't even get in the cell because they've changed the way that the drug used to get into the cell. Other microorganisms can change the way that their target looks. So if the drug targeted the ribosome, they change the ribosome. They change the shape of the ribosome so the drug can't bind to the ribosome anymore. And others will inactivate the enzymes. So some microorganisms produce enzymes that will degrade the enzymatic activity of the antibiotic. So if the antibiotic was supposed to mess with peptidoglycan synthesis, if you cut up the antibiotic, then it will no longer be effective. So how do we prevent drug resistance? Limit drug use. So do not overprescribe antibiotics. Proper drug use. So don't give an antibiotic for a viral infection. And this is really hard in pediatrics because parents obsess over their children. They want their children to be well. And if the child has something like an ear infection, they go to the doctor and they want to leave with something. They want to leave with some drops or something to make the child feel better. Well, most ear infections are viral. Unless you see gunky pus coming out of the ear, most of them are viral. So by giving a drug, it's really not going to do much for the infection. It's a virus, and the virus has to take its course and leave. But parents demand drugs, and then they end up using improper drugs. Narrow range antibiotics. We talked about that in the beginning of this lecture. So we want to target the specific microorganism that's causing the problem. And then multiple drug treatments. That's what I talked about with malaria. So today, instead of just treating malaria with one drug, you actually give 2 drugs at the same time. Because if there is resistance, it's highly unlikely that it'll be resistant to both drugs that you're giving in combination therapy. So if we start using 2 drugs at the same time to treat certain infections, that might decrease the survivability of the microorganisms, because it's unlikely that one microorganism is going to be resistant to both drugs that you give. So antibiotic susceptibility testing, I'm going to talk about at length once we get to the lab period. But questions to think about. Is every bacterial culture tested for susceptibility? No. A lot of doctors don't order susceptibility testing. That's something that they have to specifically request when they order a culture. If not, how do we determine which ones are? Well, any one that comes back where the drug's not working, then automatically we should test for susceptibility. Today, in my opinion, every strain should be tested for susceptibility, but that's just not feasible. So critically ill patients, infections in areas of the body that are critical, so like the meningitis infections or brain infections, vital organ infections, those should all be tested for susceptibility. Wound infections that look like they're spreading and becoming deeper, that may lead to an amputation or some type of sepsis, so that's a place. But it's really up at the discretion of the doctor to determine which ones are supposed to be tested for susceptibility. How is the susceptibility test performed in the clinical lab? Well, now we have this VITEC system, and I'll talk about VITEC when we get to diagnosing infections. How is susceptibility test performed in our classroom lab? In the classroom, we use a Kirby-Bauer method. Kirby-Bauer method is still used in some clinical labs, and it's the standard for a lot of clinical labs in other parts of the world. But in the United States, now we've kind of switched to the automated VITEC system. Which procedure is better and why? Well, VITEC is extremely expensive. It's very accurate and can give you results in a very short period of time, but it has its expense. Kirby Bauer is cheap, it can be performed everywhere, but it takes a long time and you have to have enough of the microorganism growing in order to be able to use that test. So the Kirby Bauer technique, an antibiogram is a profile of antimicrobial sensitivity. So it's an idea of which antibiotics will be effective against the microorganism and which ones will not. Specifics of the test that must be met. So the agar type and thickness. We use Mueller-Hinton agar at 4 millimeters thick. We have to use that agar because it allows for the diffusion of the antibiotic through the agar, and this is something that you'll see in lab happening. Thickness has to be 4 millimeters thick. If it's too thick, then the antibiotic won't diffuse enough. If it's too thin, then the antibiotic will diffuse too much. That's going to affect the test result. If being susceptible, in other words, if the antibiotic works to kill it, if being susceptible is positive, then having too thick of agar will lead to false negatives. Having too thin of agar will lead to false positives. A false negative is a negative result that should be positive, and a false positive is a positive result that should be negative. And we'll discuss more of this when we get to the lab period. Antibiotic concentration. So the antibiotic discs come from a manufacturer at a very specific concentration. If the antibiotic is too concentrated, it's going to have too much diffusion, and that's going to create false positives. If the antibiotic is not concentrated enough, it's not going to have enough of diffusion, and that's going to create false negatives. OCD, original cell density. The OCD is standardized, so they have Kirby Bauer specifics for McFarland standard units. So they say this many cells should be plated. If too many cells are plated, then the likelihood of some of those cells being resistant and the safety in numbers will decrease the effectivity of the antibiotic, leading to false negatives. If there's not enough cells, then they're going to be more easily killed, so it's going to lead to false positives. The incubation time. Incubation time should be between 16 and 18 hours. If you let it grow for too long, the resistant ones may start to grow back toward the disc, decreasing the zone of inhibition, leading to false negatives. So here's what we'll see. We put the antibiotic disc on a plate of bacteria. The antibiotic starts diffusing out. And then if the antibiotic killed the bacteria, you'll see the zone of inhibition. And then we're going to measure the zone of inhibition in millimeters to determine what that measurement is, and then we'll compare it to our chart to see if it's resistant or susceptible. So here we see an antibiotic zone of about 28 millimeters. If it was resistant, it would have to be less than 17 millimeters. If it was susceptible, it would have to be greater than 22 millimeters. So because it's greater than 22 millimeters, this bacteria that was plated on this plate is susceptible to enrofloxacin. Now we go through and measure each disc, and we can say which one's susceptible, which one's resistant. If it's a measurement in between susceptible and resistant, we call it I for intermediate. So here's a table of some different drugs and their zones of inhibition that's required to be classified as susceptible or resistant, and then anything in between would be intermediate. There's other tests that we do, like the MIC, minimum inhibitory concentration, that determines what concentration of antibiotic is required in order to clear the infection. The E-test, this is an E-test, so there's various concentrations of, let's say this is fluconazole for Candida albicans infection. So there's various concentrations listed here, and it can tell you at what concentration is required to clear the infection. And then the Vitek will not only give you different antibiotics to use, but also tell you what concentration is required in the patient in order to be effective. So the Vitek spits out results, 10 different antibiotics that are effective against the pathogen, and what concentrations of each antibiotic are required to clear the infection. So it gives the doctor options, and the doctor can use the information about the patient health to determine which is the most appropriate antibiotic to use. So if it says you need 100 milligrams of penicillin, but you only need 1 milligram of vancomycin to clear that infection on that patient, and the patient also has a penicillin allergy, well, you're not going to use penicillin if they're allergic to it and it requires 100 milligrams of the drug. So you'd go with vancomycin. So you use the patient's information and health status and all that type of stuff to determine which antibiotic to use. But with this VITEC test, it gives you a lot of different options and tells you a lot of information.